Kim Ham is going to present his work on oncogenes outside chromosomes. Kim Ham is a PhD candidate student at Stanford University. His research focuses on the regulation of the gene expression and organization of the genome in disease and development. His recent work with Howard Chang at Stanford aims to uncover mechanisms of oncogene upregulation on extrachromosomal DNA in human cancer. So, Kim Ham, feel free to start our presentation.
Thank you for the introduction, and thank you to all organizers for inviting me to give this talk. Today, I'll tell you the story about oncogenes outside chromosomes. So the cancer genome undergoes extensive changes throughout the development of a tumor. And this results in a genome that is so messed up that even using old cytogenetic tools in the 1960s, people were able to see all these chromosomal abnormalities in cancer cells. So this is a paper that was published in 1965, where the authors looked at the chromosomes of a cancer cell, and they saw, first of all, a wrong number of chromosomes, but also they saw these extra bits of DNA. And so throughout this talk, I'll focus on these extra bits of DNA, or called extra chromosomal DNA, or ecDNA. They were classically called double minutes because you often see these paired dots on metaphase spreads. In that first paper that described these molecules in 1965, the author made a number of observations that turned out to be true. First of all, the number of these ecDNA molecules varied from cell to cell. So different cancer cells have different copy numbers. They also don't have centromeres, and they appear to be DNA rings. So over the next decades, people have used more advanced imaging techniques to look at these molecules, and using DNA FISH, for example, fluorescence in situ hybridization, people were able to find many oncogene sequences that are on these ecDNA molecules. So these are a few examples showing labeling of chromosomes in blue, and you can see these red fluorescent signals for different oncogene sequences outside chromosomes. So the MYC oncogene, which is a transcription factor, cyclin D1, which is a cell cycle regulator, and EGFR, which is a growth factor receptor. And so in more recent work, people have also found that ecDNA is very common in cancer. Using whole genome sequencing data of tumor samples, like the tumor or the cancer genome atlas, people were able to detect ecDNAs in many different cancer types, including brain, ovarian, skin, colorectal cancers, and so on. And so overall, the current estimate is that across different cancer types, about 14% of tumor samples have ecDNAs. Patients that have tumors with ecDNAs have worse survival than patients without ecDNAs, or even compared to other types of oncogene amplifications, patients with ecDNAs have worse survival. And then when you look at individual cancer cells, you see somewhere between 5 and 200 copies of ecDNA per cancer cell. So not only do you see high levels of copy number amplification, there's also a high level of copy number variability between cells. So all of these observations suggest that ecDNA is pretty common in cancer, and there might be some behaviors that are different from chromosomal DNA. Yet we have very limited understanding still of some of the pretty basic aspects of ecDNA biology, such as the arrangement of DNA sequence. So what are the DNA sequences that are on ecDNA? How are they arranged? And also, how are these molecules inherited by dividing cancer cells? So in today's talk, I'll briefly talk about my work in the past few years on these two areas. So to first look at the sequences of ecDNA in cancer cells, we developed a tool, or rather we adapted a tool for isolating ecDNA molecules directly from cancer cells. So the basic idea of this method is we use we take cancer cells and we embed these cells in agarose to protect the integrity of the DNA content. And then we use CRISPR-Cas9 to make a single cut. Oh, sorry. And we use CRISPR-Cas9 to make a single cut on the circular ecDNA. This single cut will alter the migration pattern of these molecules in post-field gel electrophoresis, and we can use it to isolate these molecules from cancer cells. The same cut made on chromosomal DNA would result in two very large fragments that would migrate very differently from the ecDNA. So in this way, we can separate the chromosomal DNA from the ecDNA. Separately, we can also make two cuts outside of the amplified region, and this would isolate only the chromosomal DNA fragments, and we can directly compare the chromosomal DNA to the ecDNA within the same sample this way. So using this method, we first did short read sequencing to look at mutations that may be present on ecDNA compared to chromosomal DNA. And this sample from a glioblastoma tumor, what we found was that there's an activating deletion in the EGFR oncogene. So this is a well-characterized deletion which removes the extracellular domain of the growth factor receptor and puts it in a constitutively active state. And we found this deletion only on the ecDNA and not on the chromosomal DNA, suggesting that this might be either selected extrachromosomally or was formed after the formation of ecDNA. We also looked at single nucleotide variants. So using bulk whole genome sequencing, what you often see in these copy number amplified regions is you see bimodal distribution in terms of the frequencies of these mutations. And previously, people infer that mutations are very abundant are likely to be on the copy number amplified regions, and the mutations are less abundant, so these low frequency mutations are more likely to be on the native chromosomal locus, so not copy number amplified. What we found was that that's mostly true, except a few of these mutations that were actually accumulating on the ecDNA, suggesting that these might be more recently developed subclonal mutations. Using this method, we were also able to look at epigenetic modifications. So as a proof of principle, we looked at DNA methylation, so CpG methylation using nanopore sequencing. And so again, separating the ecDNA from the chromosomal DNA from the same glioblastoma sample, we were able to look at DNA methylation levels using nanopore sequencing. And here I'm showing the ratio of methylation frequencies of the ecDNA compared to chromosomes. So this shows that there's actually a decrease in methylation frequency at the EGFR gene promoter on the ecDNA compared to chromosomes. And this was really interesting to us because it suggests that a decrease in methylation might be one of the mechanisms for upregulating gene expression on ecDNAs. And we're also able to isolate different types of ecDNA species because we're using gel electrophoresis, which can separate molecules that are different sizes. We can then reconstruct all of these different ecDNA species using sequencing data. So here are examples of 3 different ecDNA structures that actually coexist in the same cancer cell population. On the left, you see the structure with FGFR2, but also a few other fragments coming from chromosome 10, all ligated into a circle. In the middle, you see a different circle with fragments coming from chromosome 8 and chromosome 11 containing the MYC oncogene. And then on the right, you see another smaller circle that's about 277 kb and also contains FGFR2. So this shows that there's actually heterogeneous ecDNA structures coexisting in the same cancer cell population. So we can look at the oncogenes that are present on different ecDNA species, but we can also look at non-coding regulatory elements like enhancers. And we're interested in enhancers because they they're known to interact with gene promoters to modulate gene expression. So we wanted to look at how these enhancers are arranged in relation to oncogenes. And so here are two different examples that we think are pretty interesting. We're showing sequencing data using ATAC-seq and also ChIP-seq for BRD4 and an active histone mark, H3K27 acetylation. So what this is showing is active enhancers that are present on DNA. So a peak suggests that it's accessible and it's marked with enhancer markings. What we find is that this enhancer actually is rearranged and ligated to both the MYC oncogene and the FGFR2 oncogene on different ecDNAs in the same sample. So what this suggests is actually this enhancer might be hijacked by both oncogenes and might affect gene expression. This is an even more interesting example where we were able to isolate an ecDNA that has no oncogenes. This ecDNA has only enhancers, again, marked by ATAC-seq peaks and BRD4 and H3K27 acetylation. So, so just summarizing all of the findings we saw in this work with reconstructing ecDNAs, we found numerous examples of multiple heterogeneous ecDNA species, where in some of them, a full locus from the original oncogene locus can be amplified, containing both the gene sequence and non-coding regulatory elements like enhancers. We also find examples where the enhancer from a different locus can get ligated to the oncogene on the same circle, and is likely being hijacked for upregulating expression. We also find these weird specialized ecDNA species which only have oncogenes or only have enhancers. So this suggests this analysis was very interesting to us because it suggests that there's some level of diversification of ecDNA structures in the same cancer cell population. And so we wanted to ask whether these different species of ecDNA can interact with each other. In a different study, so this is work that was published around the same time with Howard Chang, Katie Yost, Frank Shi, and James Liu. We show that different ecDNAs can interact with one another in what we call ecDNA hubs. So we think these clusters of ecDNA bring enhancers and promoters into proximity, allowing them to interact with each other. So when you look at nuclei of cancer cells with ecDNA, you actually see clusters of many copies of ecDNA. So you see all these molecules in proximity, and these clustered molecules have higher transcriptional activity compared to both the native chromosomal locus and non-clustered molecules. So we think these heterogeneous ecDNA structures can encode different oncogenes, but also carry not different enhancers and maybe other non-coding regulatory elements. And intermolecular interactions can allow them to further upregulate oncogene expression in cancer. Okay, so moving on to the second part of my talk, I'll talk about the inheritance of ecDNA by dividing cells. So the central idea of cancer genetics is that chromosomal DNA changes are inherited by dividing cancer cells, right? So for example, if this gray cell divides and acquires a mutation and becomes this green cell, all of the progeny of this green cell will have this mutation. And then let's say at some point, another cell acquires a second mutation, all the progeny of that cell will have both mutations. But this becomes trickier when we look at ecDNA sequences because ecDNAs don't have centromeres and they're unstably inherited. So this is a slide. This has been known for decades, but this is an example from a paper in 2017 showing DAPI staining of DNA, and you can see lack of centromeric signal on ecDNA molecules. So what this means is that as the cells divide, you can end up with different copy numbers of ecDNA in each cell division. So let's say this other gray cell acquires an ecDNA and now is marked in brown. The ecDNA after replication can go to one of the two daughter cells. And so one daughter cell can lose an ecDNA. And then over subsequent cell divisions, different daughter cells can have different copies of ecDNA. And so you can end up with this cell population with very, very different copy numbers. And so we asked a pretty simple question, which is knowing that there's these heterogeneous structures of ecDNA and that they interact with each other, how are these different ecDNA species inherited by dividing cancer cell? So you can imagine that if a cell needs, let's say, two different oncogenes for rapid proliferation, losing one ecDNA would be a huge selective disadvantage. So the question is, how are these different ecDNA species inherited? So to address this question, I worked with Josh Lang, Paul Michel, Matt Jones, and Ivy Wong on measuring ecDNA copy numbers in dividing cancer cells. So the way we did this was we used immunofluorescence labeling to look at a mitotic protein called Oncogenes B, which marks daughter cells coming from one dividing cancer cell. And we can look at these daughter cells and do DNA FISH to look at copy numbers of different ecDNA species. So this is an example of a stomach cancer cell that has divided into two daughter cells with FGFR2 and MYC ecDNAs. And so we can count all these ecDNAs in daughter cells and look at the copy numbers. So if the two ecDNAs are unrelated, their copy numbers in dividing cells between the two different daughter cells should also be unrelated. But what we found was when we looked at hundreds of these daughter cells, the fraction of MYC ecDNAs inherited by one daughter cell is correlated with the fraction of FGFR2 ecDNAs inherited by that same cell. And so this suggests that these two ecDNAs are co-segregated. What that probably also means is that when we look at individuals' cancer cells across the entire population, we should also see copy number correlation in individual cancer cells. So to look at that, we did single cell sequencing. And as a positive control, we looked at genes that are on the same ecDNA molecule. And as expected, we found that they're very highly correlated, which makes sense because there's the same molecules. But when we looked at genes that are on different molecules, we saw lower but still very significant correlation between the copy numbers of these genes. So in individual cancer cells, these genes that are on different ecDNAs have a correlation coefficient somewhere between 0.3 and 0.6. And then as a native control, we also looked at genes on different chromosomes, two different chromosomes, and expected they're not correlated. So what this shows is that this is not some artifact due to sequencing quality or other reasons. And so all this shows is that ecDNA species can be co-segregated during cell division, and they're correlated in copy numbers. So the next question we asked was, will these different ecDNA species respond to changes in selective pressure in a coordinated manner? So to address that question, with Brittany He, a technician in the lab, we looked at these stomach cancer cells that have FGFR2 and MYC ecDNAs. The FGFR2 ecDNA encodes the FGF receptor, which can be inhibited by a drug called pemigatinib. So we treated these cells with pemigatinib and asked what happens to the copy numbers of ecDNAs over time. So we measured the copy numbers of both FGFR2 and MYC ecDNAs over time. In the first 2 weeks, what we saw was the copy number of FGFR2 ecDNA decreased, which makes sense because the high copy number is driven by selective advantage. When we remove the selective advantage, we select over time cells with lower copy numbers. But what was unexpected, or I guess it is consistent with the idea of ecDNA co-segregation, is that MYC is also reduced in copy number, even though the MYC ecDNA is not directly targeted by the drug. But then over time, when we treated these cells with pemigatinib over a course of 6 weeks, the MYC ecDNAs eventually came back and became even higher in copy number compared to before. And so what this suggests is that the MYC ecDNA is probably under selective pressure, so it's probably being selected for, but it was it reduced in copy number initially because it was guilty by association. It was co-segregating with the FGFR2 ecDNA, which was reduced in copy number. And to ask how the MYC ecDNA can first decrease and then increase, we wanted to ask whether there are large structural changes in the genome. So to answer that question, we did metaphase spreads of these cells at the end of the experiment. And what we found was that in the control, the FGFR2 and MYC ecDNAs remain separate ecDNAs, but in the pemigatinib treated cells, the FGFR2 ecDNAs actually jumped into the chromosomes. So we found a lot of chromosomal copies of FGFR2 where MYC ecDNAs remain extrachromosomal. So this suggests that one mechanism for uncoupling two different ecDNA species might be to put one ecDNA species in chromosomes. And so we finally asked, can we try to recover the co-segregation of ecDNA species? So to address the question, we introduced another treatment where we treated the cells with a short period with pemigatinib, so 2 weeks with pemigatinib, and then we removed the drug for 2 weeks. And then we wanted to add it back and see what happens. So in the first 2 weeks, again, you see coordinated depletion of both ecDNAs. And then when you remove the drug, what you see is recovery of both ecDNAs. So you see an increase in copy numbers of both ecDNAs. And then when we added the drug back, amazingly, the MYC ecDNAs became sensitive again to pemigatinib treatment. So what this shows is that the property of correlated copy numbers can be recovered probably in very short period of time. So this is only a 2-week period of time where there's no drug. And the two different ecDNA species can become coupled again. And so to summarize all these findings, we think that cancer cells have multiple ecDNA species that can co-segregate during cell division. These ecDNAs are co-depleted when they're under selective pressure. But one way to uncouple these different species is by putting one species on chromosomes and selection of different ecDNA species. So you see this very dynamic copy number changes over time in cancer cells. So as a summary, what I've shown today is that in cancer cells, there's a lot of heterogeneous ecDNA structures that are coexisting in cancer cell populations. And that because of these intermolecular interactions between different ecDNA species and their coordinated inheritance in cancer cells, there's these unintuitive dynamics that are given rise to by the ecDNA collective behaviors. And finally, we really don't understand what conditions favor extrachromosomal versus chromosomal copies of oncogenes in cancer. And that's something that should be worked out in the future. And so I'd like to end this talk with a tribute to Barbara McClintock, who of course discovered transposable elements and won a Nobel Prize for it. And in the 1980s, she published a paper where she talked about the significance of the genome as a highly sensitive organ of the cell. It senses unusual and unexpected events, responds to them often by restructuring the genome. And so in her view, the DNA is not a static fixed entity, but is rather dynamic and responsive to environmental changes. And I think we're seeing more and more examples of this in cancer genomes. And so I'd like to end by acknowledging my collaborators, Howard Chang at Stanford, who's my PI, Katie Yost, Frank Shi, and James Liu, who co-led the ecDNA Hub paper with me, Jens Lubeck, who did a lot of the DNA reconstruction work, Matt Jones, Ivy Wong, Brittany He, and Josh Lang, who worked with me on the co-segregation of ecDNAs. And so if there's any time left, I'll be happy to take questions.
Thank you, King, for your wonderful presentation. So I have a question. When are these ecDNA appearing or being generated in the tumor life? Is it an early event? Can be a driver event?
It might be different between different cancer types. I think in some cases, it's been suggested to be an early event. And it's also been suggested that a single catastrophic event can lead to shattering of DNA fragments and ligation of multiple fragments into a circle. So it likely doesn't take a lot of time to form ecDNA circles. But I think we still need to do a lot of work to really answer that question. That's an important question.
Okay, thank you, King. We also have a question from Hannah. She says, in practice, how difficult is to distinguish quantity and regulatory interactions of ecDNA from chromosomal DNA?
Oh, sorry, can you repeat that question?
Yeah. In practice, how difficult is to distinguish quantity and regulatory interactions of ecDNA from chromosomal DNA?
Oh, so interactions between ecDNAs and interactions between chromosomal DNAs, is that the...
Oh, Hannah, you want to clarify something maybe?
Yeah, sorry.
Yeah.
I would like to know how difficult is like methodological thinking, how difficult it's to distinguish like those different DNA and all the regulatory interactions of they both.
To be honest, extremely difficult because traditional methods like Hi-C or other chromatin capture methods look at all of the DNA in your sample, right? So because ecDNAs have sequences that also are present on chromosomal DNA, all times it can be difficult to see to separate the signals. In the studies that we did that I presented here, we focus specifically on sequences that are non-overlapping. So for example, if we're looking at interactions between two different ecDNA species, we generated subclones of cells that only have one sequence on one ecDNA and one sequence on another ecDNA, and then we look at the interaction. Or in other cases, if we have different mutations on the ecDNA compared to chromosomes, we can use the mutations to distinguish, ecDNA chromosome interactions or other types of interactions. But usually it's very difficult. We need a lot of prior knowledge in order to do this kind of analysis.
Now we have a last question from Matthew Coelho. Is it possible that you are selecting for a subpopulation of cancer cells with the correct constellation of ecDNAs rather than actually selecting their gain or loss with drug?
Yeah, so I think that's part of the equation, like having the right ratio of different ecDNA species is probably important. And we know this because you see cells amplify copy numbers up to a certain number, and then it stays relatively stable, at least in the time course experiments that we're doing here. So for example, we see a mixture of ecDNA species that are relatively stable in ratio over time. And so there's probably something to the specific constellation, which is a good word, of ecDNA species, rather than just absolute copy numbers.
Okay, thank you, King, again. Thank you to both of our speakers